Hello, thank you very much for your interest in our study. I'm Jinjing Han from Santa Clara University. Today, I'm going to present our study on skin conductance synchrony during individual media viewing as a function of media valence, arousal, and emotional change rate. For decades, media psychologists try to understand information processing during media use in an individual viewing context, but we rarely looked at the links or similarities between individuals' processing patterns. In other words, we wonder if media can make us think alike and if media can make us feel alike in context of individual media consumption. The goal of this study is to understand the degree to which media can synchronize individuals' skin conductance activity during video viewing as a function of the three message variables as we listed above. Therefore, synchrony in this study refers to interpersonal synchrony, in particular, skin conductance level synchrony, which is defined as the temporal similarity between two individual viewers' skin conductance activity. Researchers have shown that media is able to generate neuron, physiological, and behavioral synchronization across individuals when they view or listen to media alone. However, the types of information in media that leads to increased or decreased synchronization remains unclear to us. We proposed that skin conductance synchrony in solitary video viewing is caused by structural and emotional content in video messages. The theoretical foundation for this investigation comes from the nonlinear dynamic system theory and from the LC4MP perspective. First, the prediction from the nonlinear dynamic system theory is that reducing the degrees of freedom in a system decreases complexity and increases order, i.e. synchrony. The dynamic system in the solitary viewing context is a human medium message system that consists of a human, a message, and a media embedded in a place. Thus, in this system, the similar media viewing environment and same media content reduce the degrees of freedom in the system. Second, from the LC4MP perspective, researchers have shown that both structural features such as cuts, pacing, camera and voice changes, and emotional content in media elicit automatic attention and emotion responses, which are indexed by predictable changes in skin conductance. Those response changes should be dynamically aligned because they are automatically elicited by changes in the message. Therefore, individuals viewing the same perceptual information from media should demonstrate a higher synchrony level than those not viewing the same media content. Unlike previous synchrony research that only focuses on synchrony strengths between individuals, we investigated not only the strength of synchrony, but also the deterministic structure and stability of interpersonal synchrony. From the nonlinear dynamic system theory perspective, we not only wanted to understand what patterns a system can exhibit, but also how patterns change over time. This leads us to examine the extent to which the synchrony patterns are deterministic, meaning the observed patterns are driven by the system itself rather than by randomly occurring processes, and the extent to which the synchrony patterns are stable in terms of how long those patterns endure. Based on previous literature, we examined the three effects on synchrony strength, determinism, and stability. Specifically, we investigate message valence effect, message arousal effect, and emotional change rate effect on those three dependent variables. So for experimental design and participants, each participant watched 8 NCAA basketball games in total. In each game, the home university, Indiana University Hoosiers, is always one of the teams. Therefore, for games that IU leads and wins, those are categorized as positive games. Otherwise, those are negative games. For games that have close scores, they are arousing games. And if they are lopsided, then those games are calm games. Each participant was randomly assigned to one of the emotional change rate conditions. Thus, emotional change rate is a between subjects variable. To test our synchrony hypothesis with as large a dataset as possible, we paired each participant with all other participants in the same rate of emotional change condition. Therefore, the between subjects variable emotional change rate is also a between dyads variable. This gives us 490 dyads for further analysis. We collected skin conductance data from the inner side of participants' left foot. Data was collected at 2000 Hertz and averaged to 1 Hertz for analysis. And here is the one data example from one dyad. We used cross-recurrence analysis to quantify 3 dependent variables. Cross-recurrence analysis is a nonlinear dynamic analysis that calculates the dynamic similarity of 2 time series. Thus, it holds no assumption about data size, distribution, and stationarity. And thus, it preserves nonlinear and dynamic features of data. We use percentage of recurrence from this analysis to indicate synchrony strength, percentage of determinism for synchrony determinism, and max line for synchrony stability. Our full paper online has more detailed explanation about this method. So if you're interested in this method, please download our full paper from the ICA system. We use multi-level modeling for statistical testing to analyze the hierarchical data structures and also to control for the interdependence of participants' data that arose from pairing participants with all other participants in the same emotional change rate. We first tested the message valence effect on synchrony. So we predicted that individuals watching negative compared to positive messages would produce higher, more deterministic, and more stable synchrony. The results revealed a three-way interaction, and it suggests significantly stronger synchrony for negative messages than positive messages for 3 out of 4 comparisons. This pattern is the same for synchrony, determinism, and stability. Therefore, negative messages led to stronger and more deterministic and stable synchrony for 3 out of the 4 comparisons. We then tested the message arousal effect on synchrony, and the same interaction effects suggest that contrary to our predictions, synchrony was significantly stronger for calm compared to arousing messages for all the message conditions, as you can see here in the graph. And synchrony for the blue calm messages was also significantly more deterministic and stable than arousing messages. Lastly, we tested the emotional change rate effect on synchrony. For this comparison, you will compare the points between the left and right panel. So the results show that the fast compared to slow changing condition had greater, more deterministic and stable synchrony for 3 out of 4 types of messages, for calm negative, for arousing negative, and also for arousing positive messages. The patterns for determinism and stability are similar, but those 3 types of significant comparisons, as you see in this right panel here, were not all significant for determinism and stability. So for conclusions, our results show that calm messages compared to arousing messages and negative arousing compared to positive arousing messages produce stronger, more deterministic, and stable skin conductance synchrony. Rate of emotional change between messages influence the skin conductance synchrony strength, but not necessarily deterministic structure and stability. Therefore, our study suggests that media not only make people think alike, but also feel alike. In other words, media can synchronize audiences both cognitively and emotionally. And because media is able to generate such an immediate and large-scale synchronization among individual viewers, future research should examine positive and negative consequences of audience emotional synchrony, such as social cohesion, social mobilization, and online radicalization. That's all what I want to share with you about our study, and thank you very much for your attention. Finally, I would like to give thanks to my IU ICR lab mentors and colleagues for your help on this study. If you have any questions, please feel free to email me with this email address here. Thank you.